All right. Good morning, everybody. Thanks for coming back. Hopefully you enjoyed yesterday's workshop. And thanks to all of you for the engagement and the discussions that we had throughout the day. So today we'll start with Jakob Macke, who is here from University of Tübingen and Max Planck at Tübingen. After Jakob's talk, we'll have a little short break. And then we come back here for the first session, which is going to be basically three people are going to talk to us about from raw data to actionable insights. That's going to be Scott Linderman from Stanford. That's going to be Christopher Bernard from Marseille and Bing Brunton from UW. So I asked Jakob to come here. And one thing actually I want to say is that like a little bit of change from yesterday in terms of the plan for the late part of the evening, because we have to leave apparently the building by 5:15. So we'll have the panel discussions after we come back from the breakout rooms, and we will integrate the reports from the breakout rooms into the panel discussion. So that's the little bit change that we had to do because we have to leave the building by a certain time. All right, and Jakob, please take it.
All right, thank you. Yeah, thanks for the invitation and the introduction. I will talk about our work on trying to build machine learning methods that are useful for neuroscience. And in some ways, I'm picking up on David's challenge from yesterday in asking the question of how we can use machine learning methods to generate mechanistic insights in neuroscience or in brain science. So what we ultimately, or what many of us ultimately want to understand is how neural circuits implement computations that underlie behavior. And the wealth of data that's been generated in the last couple of years is providing great opportunities because it allows us to measure both behavior at high resolution as well as the underlying circuits, the different properties of the underlying circuits. But linking the two is a formidable challenge, in particular because not only do we have to bridge different scales, but also different time scales. In the case of the connectome is a static snapshot of the circuit, but behavior of the animal is highly dynamic. So it's challenging to build models that can bridge the two. And the question that we've been trying to do is sort of how we can build models that allow us to link the underlying mechanisms with behavior. And there's arguably sort of in a cartoon way, there's sort of two ways to build models. The one is classic mechanistic modeling in neuroscience, which starts off by being biologically realistic because you sort of build this model using ingredients that sort of encompass or capture your knowledge or your assumptions about the mechanisms at some level of detail. But the challenge is that for mechanistic models, it's typically really hard to get them to do interesting things, in the sense that setting the parameters of such a model such that the overall system can capture the behavior or reproduce the behavior that you're interested in is challenging or often impossible. On the other hand, there's obviously been great advances in machine learning, in particular in deep learning. And the great thing about these networks is that we have very effective ways to train them. And we can train them to do very challenging computations that are at a similar complexity as behavior might be, and at least networks that can do the same tasks that animals can do. On the other hand, deep neural networks typically forego biological realism, so to sort of get mechanistic insights from them, therefore, can be much more challenging. So the sort of high-level mission of our group is to think about how we can combine these two approaches, to combine ideas from machine learning and mechanistic models, mechanistic simulations, to build tools for data-driven scientific discovery, with a focus on neuroscience, but I will also show some examples from other disciplines, in particular from physics. I will talk about three examples. The first one is a project with Srini Turaga's group on building Connectome-constrained models. And then I will talk about two technical contributions. One is differentiable simulator, and the second one is different simulation-based inference methods. Okay, so on to the first example. In this project, which was carried out by Janne Lapperlein, who's a graduate student in my group that's jointly supervised by Srini, as well as with Srini in his group, we asked the question of whether and how we can build neural network models that capture the computations of the motion pathway in the fruit fly. And we took advantage of the kind of connectomes that were becoming available at the time, and we wanted to build a model that's consistent with these connectomes, but in some ways sort of gives us allows us to model the underlying computations. So to do that, we started off by building a very simple model of that pathway. And in particular, we built a recurrent convolutional neural network which comprises the major stages of the motion pathway, and where every neuron can be mapped onto a cell type in the fly brain. So this is a deep neural network in the way it's written down, but it's a deep neural network in which every unit has a name, because it corresponds to a particular cell type in a particular location in the fly brain. We approximated this model as being convolutional. There's various reasons for this. First one is, at the time, there weren't dense connectomes yet, so we had to make that assumption to fill in the gaps. But also, we wanted to start with the simplest possible model, and therefore we made that approximation. And in a classical deep neural network, you would then train all the parameters. In this particular case, we can read off some of the parameters from the connectomes. So if we look at a particular pair of cell types, the connectome will tell us the number of synapses between these two neurons, and in fact also as a function of spatial position. So what you typically have in a convolutional neural network is these filters. In this case, for example, we want the filter from an MR9 cell to a T4D cell. We can read off the average number of synapses as a function of the spatial offset between two neurons of that cell type. And we can fill that in terms of the weight. And we can also, because of work by Jan Funke's group, we can also have very good estimates of the synapse signs. So we can build this neural network without training it. We can sort of put in the numbers by reading them off from the connectome. We call this a deep mechanistic model. And this, in our case, it had 64 cell types, a total of 44,000 cells, and we modeled 1.5 million connections. But obviously, this model doesn't have any dynamics yet, and there's still other parameters that this model has. So we tried to build the simplest possible mechanistic model of this, and simplest possible in that case means that every neuron was modeled as a passive point neuron. There's no spatial structure, there's no nonlinear dynamics. And we assume that all neurons of the same cell type are identical. We use the simplest possible synapse model, which are current-based synapses, so that the input that one neuron gives to the other one is just a threshold linear function of the input. And that gives us the following three parameters. For every cell type, we get a time constant and a resting potential. So these are 2 parameters per cell type. And then for every synapse, we get one scale factor. For every connection, we get one scale factor, and we choose that scale factor to be a proportionality constant that's different for every pair of cell types. So each weight is proportional to the number of synapses, and the sign is read off from the connectome, but there's a scale factor. And we have order of 64 squared factor.
That's the point of clarity. How come the nonlinearity is not on the total current? Not f of sum of I of
No, we have the nonlinearity on the synapse. There's no nonlinearity in the dynamics of the single neuron. Is that? These are not spiking neurons. Well, they're thought to be mostly not spiking neurons. Apparently, sometimes they can spike. We basically took those that were available in the connectomes at the time. And now there's a lot more, and we're working on that. Well, we could, I'm not sure whether we ever put them in another place. We've actually gone away from current-based synapses for now, so I don't want to, I think the every individual piece there you can easily question and ask about whether we didn't do it in a different way. Totally fine. I'm hoping for more trouble. So in total, this gives us 734 parameters, which you could argue is a very big number or a very small number, whichever perspective you're taking. One thing that's clear is that most of the parameters are constrained by the connectome. So the connectome itself is giving this gigantic constraint. If it was like a full RNN, then you would have millions of parameters in this model. But there's still 734 parameters we have to set. So how do we set them? And we turn to task training. Okay, so one thing that, that's been very popular and successful, in particular in studying the visual system, is that if you train neural network models on tasks that the visual system is thought to carry out, then at least for the primate ventral stream, it's been found that the representation that these neural networks develop seems surprisingly similar to the representations that you find in real brains. And we can take the same approach here. The difference is here that this neural network has a one-to-one mapping onto the cell types in the fly. So the question is, what task do we take? Well, it's a motion pathway, and flies excel at detecting motion, so we take a motion task. We feed in movies. In this case, it's the Sintel movie, which is a synthetic, also sort of a synthetic movie which has the advantage that we know the ground truth optic flow at every voxel. So the task that the neural network is then trained to do is to reconstruct or predict the optic flow at every pixel. And to do that, we have our mechanistic model, and we have a decoder that takes as input all the output neurons from the motion pathway and tries to predict the optic flow at every pixel of the frame. And we can do that, and then we can train this recurrent neural network on that task. And I will point out that we didn't use any neural activity data to constrain this model. So that allows us to then ask the following question. Given this model that's constrained by the connectome, an extremely simple model of the biophysics, and this task training approach, does this model develop neural activity that looks realistic? And there's extensive knowledge on the visual system, and we can use that to compare visual system responses to our model. And we started off by looking at flash responses. So we simulated this experiment in the model, and then kind of compared model responses to reports from the literature. And what we found is for those cells that have been reported to be on-selective, and those cells that have been reported to be off-selective, they're also on or off-selective in our model. And we repeat this procedure across many models trained from random initial conditions, so we get an estimate of the variability. And at the same time, we also get predictions for every other cell type in the model, including all the cells that haven't been experimentally reported so far. The next feature that we looked at is direction selectivity. When we look in our model, we find that the T4 cells in the model are tuned to on motion. The T5 cells are tuned to off motion, which is also the case in the real fly. And we also find predictions for some other cell types that are predicted to be on or off motion selective in the model, but which don't inherit their motion selectivity from the famous T4 and T5 cells. We can look at the T4 and T5 cells in more detail. In particular, for these cell types, they have this famous cardinal tuning that they're tuned to left, up, right, and downward motion. And when we compare the best task-trained models, then they have the same direction selectivity, so each of the T4 cells and each of the T5 cells maps onto direction selectivity that's experimentally reported. I will say, though, that when we train many such models, we find variability. And one thing in particular that we find is that we find different clusters of models. So if, in this case, if we look we clustered all the T4, in this case the T4C cells, and we find 3 different clusters. One is the cluster corresponding to the best performing models, and this is the model that has the correct tuning, but we also find a cluster that has the opposite tuning and a cluster that's untuned. So in this particular case, this is the best performing cluster, and we know that this is the right cluster in some ways, but the nice thing is that if we didn't know this, we would have a mechanism to generate predictions about how these models are tuned, right? So if we had no way to tease out which of that was the right cluster, we could turn this into a prediction to run an experiment to rule out a whole set of models. And I think in general, when we fit or try to build models of systems as complex as this, we will always have to live with the fact that these models are not, that there will be large parts of the model that can't be constrained by the data or the task. So I think having a mechanism to sort of visualize the variability and use it as a ways to identify informative experiment will be important moving forward. One thing that turns nice is if you look at the input neurons for the T4 cells, then if you constrain the T4 cell tuning, that also propagates to constraints on the input neurons. So if someone did run experiments where they rule out one of those clusters, you don't have to run this for every individual cell, but sort of the constraints you will get will then propagate through the network. This is just sort of, I guess, an example of how this could be done, but I'm sort of optimistic that moving forward, we will be able to use these model-based approaches to sort of create hypotheses for which experiments should be carried out and use them to constrain much bigger models without having to record every single cell. The reviewers asked us to compare to 26 different studies, and so we did. And in that paper, there's a very long supplement of how every individual cell that has been reported for compares to the literature. I will not go through these 26 different studies, but there's a very long supplement of the paper that I encourage you to read if you're interested. All of this is published. I just want to give you a quick idea of how we are working on this, how we're extending this at the moment. When we built this model, we didn't integrate any activity measurements, which was important, I think, for this proof of concept, but in some ways, obviously, is strange, right? If you do have neural activity measurements, why not use them to constrain the model? Linda, a student in the group, is working on this. This turns out to be a very tough multitask learning problem, but we're making some progress on constraining models, both by task constraints and neural activity constraints. Sina, in particular, is working on using the model to do science, and she's asking the question of sort of what the role of different recurrent connections in the model is for different processing tasks. Felix and Isaac have been looking at other tasks. So we constrained this model by this motion estimation task, but it's unclear what properties of the task are critical for constraining the model. So we're trying to extend this on a wider range of tasks. And obviously now there's full dense connectomes that would allow us to model a much wider range of cell types and also go beyond the motion pathway. And we're busily working on building larger models that integrate this. Given the theme of the workshop, all the code for this is available. Janne also made in the process another tool he calls DataMate, sort of for configuration management for his models, and both of these are publicly available. Okay, this brings me to the end of the first section of this. So what I'm excited about is maybe not the details of this particular model, and as you saw, this in some ways we built the simplest possible model that we could think of that would be able to sort of capture things at that scale. But what I'm excited about is that I think this approach can be extended and scaled, right? We can use this to also work on other tasks. We can use this to work on other brain regions. And it might also provide a way to integrate different kind of data. At the moment, we basically only use connectomic data, but we can, I think, use this as a scaffold to sort of to attach other kind of data into such a model. The model makes a lot of predictions. Many of them will turn out to be wrong. And then we can use that as extra constraints to retrain the model. And once in a while, we'll find that however we retrain the model, they can't be captured by the model, and we have to change it. And then we will do this. So I think I'm more excited almost about the approach, how we can use this moving forward, than the particular details of the model. Okay. So the one thing that, sort of on a technical level, one thing that was critical for this is that we could optimize the simulation like a deep neural network. And so in fact, the simulation that I showed you was all implemented as a differentiable simulator. So we could take the gradient of the model output with respect to any of the parameters. And I think that strategy is something that will be important if we want to constrain such models by tasks and or by data. And in particular, we would like to do this also models that have more cellular detail than this particular model. As I mentioned, it's the simplest possible model of biophysics. So if we had a system where we wanted to build in more cellular details, how would we be able to use the similar approach? And what we would have to do is have means to optimize or train detailed biophysical models with tools from deep learning, in particular variants of gradient of descent. And this is a question that Michael Deisler, a graduate student in the group, asked himself. And he basically said, wouldn't it be great if we had a tool that would allow us to do this? And he was thinking of something like Neuron, the Neuron simulator, just in a way that it's implemented in a sort of deep learning friendly framework so that you could do things like parallelize across parameters, data, branches, and cells, and importantly, compute the gradient of your model output with respect to all the inputs. And we discussed this several times, and I basically always said, look, this is a great thing, and someone should do this, but it's going to be way too much work for like a single graduate student to take this on. And then one day, he came back from vacation, and he basically said, look, I have a pilot of this. And what he had done is he effectively went sort of very deep into the weeds of the Neuron simulator and sort of looked at the papers and how it's implemented. And there's a key step if you want to implement these things efficiently. And you need an implicit Euler solver because these equations are stiff. And one of the things that implement Neuron is a particular way to efficiently solve an implicit Euler solver on branched structures, because multi-compartment neurons are branched. And there's a way to sort of reorder the computation in a way that we can do this linear in the number of compartments. And this is the bit that Michael re-implemented from Fortran to JAX, so that this is now, when you look at this, when you run a simulation in Neuron or in Jaxly, which is the name of this tool, then you get exactly the same simulation out. The difference is that you can call it, like in the JAX deep learning framework, and it will take care of doing things efficiently in a parallel way on GPUs or TPUs without you having to sort of worry about these things. So depending on the question, this can be much faster than a CPU implementation. As sort of one example, this is a model, this is not this particular model, but sort of if you had a model with 2,000 neurons and 700,000 compartments and 1 million synapses, that would give you an ordinary differential equation with roughly 4 million states, because we use 4 gating variables for each of them. And if you did simulate this in Jaxly on a single A100 GPU, then if you did run a simulation for 200 milliseconds, discretized such that this is moving 8,000 time steps forward, that would take 21 seconds for the simulation, and computing the gradient for all the 3.2 million parameters would cost 114 seconds in this case, okay? Just because sometimes it's asked, well, can you just take your normal simulation and do finite differences? Doesn't this also give the gradient? The answer is yes. In this particular case, it would take about 2 years to get the gradient with all the different parameters, okay? So this is a tool we've built, and we've applied it to a range of different tasks to mostly demonstrate the capabilities of this. And we've looked at both sort of data fitting tasks, where we try to optimize the parameters of model to fit particular parameters, and also machine learning tasks, where you try to sort of make a cellular realistic network do different tasks. I will just show 2 examples. All the others are in the preprint. And we use this to fit models to voltage recordings from the Allen data set. This is classically a task that's done with genetic algorithms. In this case, when we do this with gradient descent, we find that sort of in our comparison that the genetic algorithms do this much more efficiently than gradient descent does this much more efficiently than genetic algorithms. These are just 4 different examples of real cell traces and the underlying sort of reconstructions from the model. A second task that we looked at is like if, I mean, this is sort of a hypothetical task, but if we did have a high dimensional, dense voltage imaging of a neuron, could we create, and we knew the morphology, could we set the parameters of such a model such that it would produce exactly the same activity patterns? And as a proof of concept, you can use gradient descent on a very high dimensional parameter space in this case to find a configuration of these parameters such that your model output or your output neuron is an exact replica of the model that you put in. We've sort of compiled these tools in a toolbox we call Jaxly, and there's an associated library called Jaxly Mech, which tries to implement a growing list of channels and synapse models so that you don't have to sort of code them up yourself when you use this. There's a scientific programmer in Philip Behrens' group that is sort of actively working on maintaining and extending this database, and we're hoping this to be a useful tool for people to sort of train their own mechanistic models, either on machine learning tasks or to fit them to particular data sets. So what I showed is that Jaxly enables this gradient-based parameter identification for detailed biophysical models. But I think it's also important to sort of point out some of the limitations and consequences that this has. The first one is it's great to have gradients, and many things get much easier typically when you have gradients, but these are still hard optimization problems, and they can be stuck in local minima. What we often do is sort of some combination of doing doing gradient descent with many restarts. One thing that's really hard is to effectively and efficiently do backpropagation through time for many, many time steps. So if you have tasks where not only you have to fit long data segments, but the information has to be propagated along through many time steps, then gradient descent becomes really tricky. And I guess the theme will also be apparent in Scott's talk, where he talks about some methods to do this very effectively, unfortunately requiring architectures that don't necessarily map onto this problem. And this sort of power obviously also comes with responsibility in the sense that if you can optimize hundreds or thousands of your parameters, then you will run into things like overfitting. or it might be challenging to interpret these solutions. So there's sort of not a magic tool to give you the model, but I think it's a good tool to really explore the range of models that can capture something. And kind of my personal take on this is whenever people say, oh, the problem is so unconstrained that we can't optimize and therefore we did it by hand, then I'm always thinking, well, but you also just found one parameter set by hand, so why should in some ways trust your hand-picked parameter set more than exploring a wide range of parameter sets that can fit the data and then trying to sort of navigate the space and reason about which different parameter settings I find plausible and whether there might be surprises, so where there might be parameter settings that I didn't think of by hand but which are a priori also biologically plausible. But these things will be needed if one wants to use these tools in a sensible manner. All right, and this brings me to the third part of the talk, which is sort of really describing a range of work that we've done over the last couple of years, which is looking at a related problem, but sort of a slightly more general take on this. So one issue that one runs in often, in particular neuroscience, is that we have some model, and it's a model that might not necessarily be differentiable. In fact, it might be kind of a black box simulator. Someone might give you a piece of code and say, with that code, I can create simulated data. If I put in the parameters, I can generate simulated data. But what I'm really interested in is which parameters I should set to kind of explain my data. And one thing that computational neuroscientists have historically done a lot is, for example, to build these spiking network simulations and then use the brute force search or gradient student search to sort of find parameters that capture the phenomenon they're interested in. And this is something that we wanted to overcome. And so what we wanted to do is develop tools, and we're phrasing this as a Bayesian inference approach, that we say we want methods that allow us to do Bayesian inference on models where we don't have access to the internal workings of the algorithms. So sort of to be a little bit more formal about this, for Bayesian inference, you need some forward model. In this case, we say a model with parameters theta, and that outputs simulated data x. And we assume that we have this black box simulator, so we can simulate different x's from it. We have some prior over parameter that tell us the parameter ranges that are plausible, and we have some measured data, which I call XO in this case. And what Bayesian inference does, it gives us the posterior distribution. And in these settings, it will often be the case that we're not only interested on some parameter set, but the whole shape of the posterior distribution. So for example, there might be different parameter configurations that explain the data equally well, and would be important to try to capture all of them, or at least as many of them as we can. All of you likely know that to compute the posterior distribution, we take the product of the prior and the likelihood, and we normalize this. The catch here is that if you just have a simulator, then that you can generate samples from this likelihood x of theta, but you can't actually evaluate the likelihood p of x given theta, right? When you generate a spiking neural network simulation, some output comes out, but there's no number that says the output that came out has likelihood 0.3, okay? And this is the likelihood you would need to compute the posterior distribution. So what simulation-based inference tries to do is it's sort of a field that tries to estimate the posterior distribution just given access to model simulations. And we focused on one particular approach in the last couple of years that I'm going to try to sort of visualize. So this approach starts off, you start off with your prior, and you start off by just randomly drawn parameters from your prior. And then for each parameter theta that you drew from your prior, you generate an associated simulation x. And when you do that, you get a long data set of parameters and associated simulations. And once you have such a parameter set, you can do what machine learning does very well, and to turn this into a supervised learning problem. You can say, given my simulated data x, can I reconstruct the parameters theta that generated it? So you train a machine learning regression algorithm to predict the parameters that generate your simulated data. And because we are always interested to do this probabilistically, in our case, we don't actually use a regression algorithm, but something that's slightly more complicated, and that is a conditional density estimator. So given simulated data x, we want the algorithms to tell us the distribution of thetas that are compatible with this. And one thing that we leverage is the fact that there's been very powerful neural network-based conditional density estimators that have become available in the last couple of years, we use normalizing flows a lot, and there's other tools such as diffusion models that can also be very handy in other situations. Once this system is trained, we can plug in the empirical data XO into this conditional density estimator. And if you do this correctly, then it turns out that under some assumptions, the density returned by this density estimator will be exactly the posterior distribution that we're interested in. So this gives us a recipe for training neural networks to learn posterior distributions without ever having to evaluate likelihoods explicitly. This approach might strike you as inefficient in the sense that you generate lots of random simulations at the beginning, including many of them that look nothing like the real data. The nice thing is there's ways to close the loop and to say, once we have an initial estimate of the posterior, we can use that estimate of the posterior to target additional simulations that are in high probability regions. So you sort of focus your simulations on regions which are plausible. And this overall approach of training neural networks to perform Bayesian inference in this particular way, we call sequential neural posterior estimation because it uses neural networks to estimate posterior distributions, and it can be used in the sequential mode where you adaptively generate your simulations. The basic idea of this goes back to approaches called regression ABC in approximate Bayesian computation. Ian Murray and George Papamacario sort of lifted this to the machine learning world in 2016, and we and other groups have extensively worked on trying to extend this to different scenarios and making these approaches more robust and efficient. I think for the user, the one thing that's important is that we and others have spent a lot of time to sort of build a toolbox that hopefully allows you to use these approaches without having to really sort of understand or be able to code up all the individual components. There's a toolbox that we've been extensively used and that we're now trying to sort of push out from the lab and make this a community effort, and I think we're making some progress for this to be a community toolbox rather than a toolbox that's just maintained by my lab. And this toolbox is now used for a wide range of different studies in neuroscience, but also in many other fields. And what I want to do for the rest of the talk is highlight two possible applications, or two applications we've worked on, and also then share with you at the end two methodological innovations that we have currently working on. So the thing I want to point out is that if you train these networks, if you train neural posterior estimation this way, then it has this property that's often referred to as amortized inference or amortization. So you spend a lot of simulations and a lot of time to train this conditional density estimator. But once it's trained, doing inference with this is very, very fast, because all you have to do is take your new observation, run it through your inference network, and you immediately get a new posterior. And this is great because, I don't know, Bayesian inference, at least sort of when I grew up, Bayesian inference was just a gigantic pain, in the sense that if you wanted to do this in a flexible manner, you basically had to do something like MCMC sampling, which meant you had to be very patient for these things to work. So it was just impossible to run this on anything that's a high throughput application or anything where you sort of want real-time answers. And these amortized inference approaches now mean that after this initial investment, you can do real-time applications and you can do high throughput applications. We've used this in a variety of different applications. For example, for identifying parameters of ion channels. In imaging, this is very useful because you often try to solve an inverse problem at very different sort of patches of your input or at very different pixels. So we've used this for super resolution microscopy. We've used it for identifying plasticity rules. There's a recent project with the group of Philip Behrens where we use this to sort of identify mechanistic models and relate them to different ion channels. And one application that we've also currently or recently worked on is to use this for diffusion MRI, where you also have an inversion problem that you want to solve at every voxel of your scan. So if you have a way to do Bayesian inference and to rapidly do this on many, many voxels, then this is something that can be very beneficial. I want to talk about two examples in a little bit more detail. And the first one, and in part because David challenged us yesterday to talk about applications on physics, is actually an application from physics. This is a simulation or an image of two black holes that merged 1.3 billion years ago, and they emitted gravitational waves that were picked up by the detectors, by the LIGO detectors. And so we get these sort of tiny gravitational waves, and we have a forward model from general relativity that tells us if we know the location of these two black holes, their masses and their spins and 10 other parameters, we would be able to predict what the gravitational wave should be picked up by these detectors. So the inverse problem, that's sort of a classical and important problem in physics, is to say, how can we invert this? Given these strain measurements, how can we reconstruct a posterior distribution over the underlying, of the properties of these black holes? And this is a question that Max Stacks, who's a graduate student with Bernard Schulkopf, and Stephen Green, who at the time was a postdoc with Alessandro Buonanno, have been working on, and they basically use simulation-based inference to tackle this problem. And what Max and Stephen developed is a variant of neural posterior estimation that he called Dingo, and this method can estimate these posterior distributions on the order of seconds, where if you used MCMC, it would take you order of hours or order of days to solve this. And one thing that's been very instructive for us is to work in these collaborations with physicists, because there's obviously a lot of beautiful symmetries and other things you can build into these algorithms, so there's sort of more bells and whistles that I've not spoken about. But also it's been an instructive experience for us to sort of look how large-scale collaborations and data sharing and coding thing work in physics, and it's quite a different world. So it's been quite very interesting to sort of peek into that world. You could still argue saying, like, why do we care about fast inference? There have been like order of 50 of these gravitational wave events that have been detected. Why is it a problem if we need to wait a few hours? And there's two reasons for why that can be a problem. The first one is there's more sensitive detectors coming online, so the number of events that will be picked up is predicted to rapidly go up over the next couple of years. And there's also a second reason, because another type of event that sends out gravitational waves are these binary neutron stars. And the great thing about the binary neutron stars is not only do they emit gravitational waves, but they also emit electromagnetic signals, light. So if you had a way to do inference on gravitational wave signals fast enough that you can point a telescope to that location in space, then you can measure the kilonova and other things coming up after the event. And the faster you can do this, the more interesting information you can glean. So this is a reason for why it's a big difference if you can do inference like a couple of days later or a couple of minutes later. And Max worked extensively on extending this approach such that it's also applicable to binary neutron stars. And using this approach, you can then estimate posterior distributions that give very good localizations, even using data from sort of the pre-merger data from the in-spiral. So you can kind of make predictions about where that event will be even before the event has happened. And that sort of buys you more time to, or would, when these things are put into production, which they're not yet, would buy you more time to sort of to point a telescope in that direction. Okay. And this is an example of a method that we really started working on in neuroscience, and then the same kind of machine learning algorithm turned out to be useful in other fields. Okay. My second example, though, I'm returning to neuroscience. In fact, I'm returning to the example that I motivated at the beginning, and this is something that Richard and Michael, Auguste, and Pedro have been working on. And this is the question that I sort of mentioned in the beginning. If you have a set of spike trains, and you're trying to fit sort of a classical computational neuroscience model to this, in this case spatially unstructured, maybe using exponentially integrated fire neurons, and you have some sort of spatial clustering. People have spent a lot of time tuning parameters such that for a particular recording, they can capture some summary statistics you might be interested in, like you want to capture the burstiness or the variability or something like this. So what Richard asked himself is, can we automate this in the sense that, can we build a tool that allows you to compute some summary statistics from your neural data, either something like the power spectrum or a few handpicked summary statistics that computational neuroscientists like to use, and can be then use one of those pre-trained density estimators so that you can rapidly estimate the posterior distribution without having to tune parameters yourself. And he called this tool AutoMind. This is one example that he applied this to. So these are organoid recordings for multi-electrode arrays. This is something he actually worked on in his PhD before he joined my group. And what you observe there is you have this characteristic changes in some of these statistics. So the interburst interval on the burst width and other properties change in a characteristic manner during development. And what he wanted to do is sort of fit mechanistic models to each of those stages and then try to come up with predictions for how the parameters of this model would change during development. So he applied AutoMine to this. He firstly found that the models could capture the rough statistics of this. Obviously, they don't capture all the details. And they also capture the development of these things over time. So the mean of the interburst interval and the coefficient of variation and the width of these things sort of change, sort of you can capture these changes over time. And then this gives you a tool for trying to inspect the underlying parameters and try to sort of reason about whether the predictions this approach makes, whether they seem plausible or not. Sort of as I referred to at the beginning, when you have when you fit high-dimensional models to data, then interpreting the parameters can be a challenge. So it's not like that this will give you an answer, but it gives you more sort of a space of hypothesis to explore. So to maybe illustrate the challenges that come up in this here is looking at the posterior distribution over two of these parameters. One is the time scale of adaptation. The second one is effectively the strength of adaptation. And is looking to the model fit at 9-week or at 32 weeks. And what you see is that basically the time scales seem to shift, and the strength of adaptation seems to be strongly constrained at the beginning and basically unconstrained at the end. Okay, so basically you find these parameter degeneracies in the sense that you might have trade-offs between different parameters, or you might have parameters that are unconstrained. And the nature of these degeneracies, what is constrained, what isn't constrained, both for single parameters and combination of parameters, actually changes in the same model for where you are, which data you fit this to. So so whenever we interpret these kind of things, we also, in some ways, have to think about the alternative explanations that are also consistent with the data. And I think that's a challenge. I don't think it's a challenge that is created by the tool. The challenge was always there, right? Just by the tool, it sort of makes it very visible that not only do you find models that fit your data, but you might find very different models that fit your data just as well. And I think that's something generally we have to grapple with, right? That at that sort of, quite quickly, it's not the case that there's a single model that might fit your data, but there's always spaces of models. So we really have to think about, reason about spaces of models, not only individual models. Okay. I'm almost at the end, and in my last couple of minutes, I want to share two methodological innovations that we've made for this, for simulation-based inference that might be interesting for applications. The first one is sort of advertise this amortized property, right? You train your neural network, and then it's very fast at producing inference on new data. And it's great, but it's actually also very inflexible, because something might change, and then you have to start from scratch again. And there's some very small things that might change. So for example, you might have missing data. Or you might only observe sort of parts of your data. Or you might fix some parameters. You might know, well, this parameter, I actually haven't, I want to clamp this to some value. So I shouldn't simulate over it, it should be clamped. So there's sort of a lot of these things that might, or you might change your prior, because you might have a new data set, and you might have additional knowledge that says, actually, I want to change and use a different prior. All of these are things that for neural posterior estimation, we just have to start from scratch, and all amortization would be lost. So what Manuel has been developing is a tool that he calls the SimFormer, which is effective neural posterior estimation, but we use a very flexible architecture, which in this case is built on a transformer, where you basically put in a long list of parameters and data, and you also tell which of those parameters you've observed, or which of those parameters and data you've observed, and what you want to do Bayesian inference over. So you can sort of at test time decide which parameters you want to clamp to what value, and using diffusion guidance, you can also actually modify your prior. So it's like neural posterior estimation, but you can delay some of these decisions to inference time. Another thing that this approach does is that you can design your transformer in a way that it respects known structure in your simulator. So what does this mean? If you have your simulator which has two components, and you know that they don't interact in your forward model, then if you can tell your inference network, it's able to learn posterior distributions faster because it doesn't have to figure out that these things don't interact. And you can design the attention mechanisms in your transformer such that it's already baked in your inference network that it knows about what these dependencies are. So long story short, what this gives you is something like neural posterior estimation with the additional advantage that you can delay some of these decisions to later, And the second advantage is it requires less training data because you can, if you know something about your simulator, you can put it into your attention mechanisms. Okay, so this is the first tool, and this is published at ICML last year. The second one, which is something we're currently working on, is to try to tackle this issue of simulation efficiency. Because the one thing I've not said explicitly is to train these neural networks, you need to train neural networks on a lot of simulated data. And that's a problem if your simulation is slow. And so what we've tried out recently is to say, well, let's not train simulators, but let's take pre-trained foundation models and use them as density estimators. In particular, there's this new foundation models for classification regression. TAP-PFN is one of them. And they're basically pre-trained. You just give it a training data set and it gives you a classification algorithm. You don't have to do any training. It turns out you can use them also as density estimators by turning this in using an autoregressive mode. So for density estimation, you can think of, I predict my next dimension, and then sort of you predict one dimension at a time. So you can turn your density estimation problem into a sequence of regression problems. And it turns out when you sort of repurpose or abuse these foundation models for conditional density estimation, you can plug them into neural posterior estimation, but you can then do inference in a much, much more data efficient manner. This is an example where we applied this to stomatogastric ganglion. The details here, don't matter, and I didn't talk about them. The thing that you should look at is this comparison between this pre-trained approach in orange and the classical neural posterior estimation approaches, which are the different shades of blue. And what hopefully you can appreciate is that you can get the same quality and for example, distance to the observations you care about using sometimes orders of magnitude fewer simulations. And I think this is exciting because it will allow us to also tackle much more expensive simulators than we've been able to tackle previously. This brings me to the end. I think overall, I'm excited about that the combination of large-scale neural measurements, better tools for mechanistic simulations, better tools for machine learning, for parameter inference, provide opportunities for building large-scale models of neural mechanisms underlying behavior. And a second part of that, in particular in the fly, I think is going to come from much better body models. Bing said she will at least partially talk about this in her talk. And I think this combination of better brain models and better body models will hopefully start a new generation of models that really allow us to do computation modeling of the circuits or the neural mechanisms underlying behavior. And maybe we get to something that's foundation models, but they're mechanistic foundation models in the sense that not only do they fit data and predict future data, but they also have statements about the underlying mechanisms and allow us to predict things like perturbations of the underlying circuits. With that, I want to thank my group members, the funding sources, and I will advertise that we have open positions both at the postdoc and PhD student level. So if anyone is excited to work about these questions or thinks they can do this much better, please let me know. We are moving into a new building where we have plenty of desk space. So, thank you very much.
All right, so we have time for a few questions. Thank you, Jacob, for your wonderful work. I'm going to start with myself, actually, with one question about your simulation-based inference. So if you have surjection in your forward model, you end up with a situation with many different configurations can lead to one set of observations. So let's take, for example, the case for ion channels, which you mentioned, right? Like many different combinations would lead to the same sort of membrane potential. And then therefore, like building a sort of a probabilistic model of what is the underlying set of configuration would be basically ill-posed. How do you deal with that?
The parameter identification problem is ill-posed. The problem of estimating the posterior distribution is not ill-posed. So this is actually exactly one of the main reasons why we're interested in getting the full distributions, because they give us all the parameter configurations. We've explored this in work that I didn't talk about that used the stomatogastric ganglion as an example, because that example, obviously, in neuroscience has been famous for exactly that kind of degeneracy. And when you do this, you find that the network finds multiple solutions that produce the same data. We then also went on to analyze the structure of these solutions. As far as we can tell, when you find these different solutions, there's always a ridge between them. So as far as we can tell, they're not islands in this case, but they're sort of more these elongated ridges of parameters that still you could interpret as multiple solutions. So I think the different They're different in several ways, so let me sort of try to pick one. I mean, the first one is in terms of the applicability. For gradient-based methods, you need the ability to compute gradients. So this is something where you can't use SBI. In terms of what your underlying goal is for gradient-based methods, at least classically, the goal is to get a single parameter estimate, whereas for simulation-based inference, the goal is to get a posterior distribution. Having said this, when you have gradient-based methods, you can just do gradient-based methods for multiple initial conditions, and you still get sort of some ensemble of solutions that you can interpret as not the posterior distribution, but at least gives you some measure of the variability. And these gradient-based methods often also turn out to be useful as a building block within a bigger Bayesian inference framework.
All right, last question.
So regarding the last part of your talk, is it possible to try to estimate which types of models are going to be better, to try to do a search through the space of models which are going to allow for more interpretable posteriors which are going to be present? Just to give an example, we have neuronal models which are biophysically detailed, which are super degenerate, just as you said, or there are glyph-type models which have been built to be convex optimization between events, and in some cases, one might be just easier to search for the other. Can you search through the space of models to do that?
I think it's a very interesting challenge. We did one study where we extended simulation-based inference to simulation-based model discovery. The approach we came up with at the time was not very scalable. We are currently working on hopefully better and more powerful approaches, because I personally think this is a super interesting question, because as you probably agree, finding the right model to begin with is a big challenge, which is maybe even more important than a parameter inference challenge. So we're trying to extend these approaches for exactly this, but I don't know, maybe next year. But we don't have results yet.
All right, let's thank Jakob one more time.